Hello, this is day one of explaining something to understand it better, also known as the Feynman technique. Today's topic is next generation sequencing, NGS in short. So, NGS is a technique that is used to get to know the sequence of bases in DNA or RNA. So, DNA is made up of four bases A, T, G, C, adenine, thymine, guanine, and cytosine, and RNA is also made up of four bases AUGC, adenine, uracil, guanine, and cytosine. So to know the sequence of these bases in DNA or RNA, we have to do NGS. In this video, I'll explain 1. What we do before NGS, 2. What we do during NGS, 3. The different types of NGS instruments or machines that are used, and 4. What we can do with the DNA and RNA sequence, the data after NGS. So these are the steps that we do before NGS. After getting the sample, the cells, at desired density, growth stage and quantity, the first step is cell lysis. Since you want something that is inside a cell, the first step is to break open the cell. The second step is separation of debris from lysate. Debris is something that you don't want, like a big protein, the cell membrane, etc. And lysate is something that you want, DNA or RNA. Third is removal of contaminants. So even from the lysate, you will have to remove some contaminants. Fourth is precipitation of nucleic acids. From the remaining mixture, you precipitate out what you want, nucleic acids. And then you wash and store. Now I'll explain each of these steps in detail. So the first step is cell lysis. There are three different methods to do this. One, chemical. Here you use detergents, pleotropic salts, or phenolics. All three work in a similar way where they target and damage the cell membrane and proteins. I've given examples for each. Two, enzymatic. Here we use enzymes. For example, proteinase K, which is a serine protease that digests proteins and enzymes that can degrade DNA and RNA. So since we want to work with DNA and RNA, we want to preserve it. So anything that degrades DNA and RNA, we degrade it first. Calcium ions are required for maximum activity of proteinase K. Lysozyme and lysostapin are two other enzymes which work by breaking a bond in the cell wall of gram-positive bacteria for lysozyme and staphylococcus for lysostapin. Lysostapin is a zinc endopeptidase. 3. Mechanical. The first method here is bead beating. Here you take the sample, you take beads and you take lysis buffer and you put these three things in a bead mill. The bead mill moves these three things at very high speeds. This results in the beads and cells colliding, which breaks open the cell walls or membranes. Second method is the French pressure press. So here the cell suspension is placed in a pressure chamber and a hydraulic press applies high pressure. So this is something like a syringe. Imagine a syringe, a liquid inside the syringe. You press the syringe from behind, which generates high pressure, and through a very small opening that liquid comes out. So similarly here, the cell suspension comes out of a very small pore, which results in breaking the cell walls. But both these methods are not used because bead beating results in breaking of DNA or RNA also that we actually desire, and French pressure press is an obsolete technique. So the third technique is used, which is ultrasonification or cavitation. I think you cannot see this properly because of my face, but I'll ensure to explain it properly. So in this technique, a probe or a sonoprobe is put in the cell suspension and this emits high frequency ultrasound waves, 20 to 50 kilohertz. These ultrasound waves create high and low pressure in the cell suspension. So when there is low pressure, bubbles form and they become a little unstable after some time. And when there is high pressure, these bubbles burst open, which releases a lot of force, high temperature, high pressure, and due to all of this, the cell membrane is disrupted. So even after you break open the cell, there will be components that are useful to you and components that are not so useful to you. So this is what the second step speaks about, separation of debris from the lysate. We use clarification, which is removal of the insoluble contaminants like cell debris and unlysed cells by centrifugation filtration. So these are physical methods which can remove insoluble contaminants. Step 3 again is removal of contaminants. This is where you remove the soluble contaminants. So DNase, RNase or proteinase K treatment is used. DNase is something that degrades DNA. So you will use it when you want to work with RNA. Similarly, RNase is something that will degrade RNA. So you will use it when you want to work with DNA. The fourth step is precipitation of nucleic acids. To do this, we do something called vortexing. In vortexing, we take the nucleic acid and we add salt and we add alcohol. Now why do we add salt? Salt has Na plus ions, nucleic acid has PO4 3 minus ions, Na plus and PO4 3 minus ions neutralize each other. This is important because this makes the nucleic acids less hydrophilic, that means less water loving. Since we want to take the nucleic acid out of water, we want to separate it from the water, we want to make it less water loving. That is what Na plus ions from the salt allow us to do. Second is, why do we add alcohol? Alcohol has a lower dielectric constant than water. So interaction between Na plus and PO4 3 minus, like I explained in the first step, this depends on the dielectric constant of the medium. Water is polar, that means it has a high dielectric constant, which allows Na plus and PO4 3 minus to remain soluble. So the DNA remains soluble. But alcohol is non-polar, so that means it has a low dielectric constant. This disrupts the solubility and therefore there is less hydrophilicity of the nucleic acid and high precipitation. Then you have to wash the DNA. So the first step is to wash the DNA pellet with 70% ethanol multiple times to remove any other contaminants or reagents that we have used during the process of DNA isolation. Then we air dry it to remove all excess ethanol, all ethanol. Otherwise, in the further steps, ethanol may interfere. And then we put DNA in ultra pure water or tris EDTA. So tris is a buffering agent which maintains stable pH for DNA stability, and EDTA is a chelating agent which binds to some ions like Mg2+, which are important as these ions are cofactors for nucleases, enzymes that degrade DNA. So by binding to these ions, EDTA protects DNA from degradation. And then you store the DNA. So for immediate use, you store it at -4 degrees, for short-term use at -20 degrees, and for long-term use at 80 degrees, -80 degrees. And many freeze-thaw cycles can damage the DNA. That means removing DNA again and again and using it for experiments and then freezing it again. So that is why it is divided into batches, and a batch contains that amount of DNA that is sufficient for an experiment. This process is known as aliquoting. It divides the DNA into smaller batches with just enough DNA for a single experiment or use.